Hello dear students. In my last videos, we have completed chapter number 5, Morphology of Flowering Plants. Today, let us start with a new chapter, that is Anatomy of Flowering Plants, from Unit 2, Structural Organization in Organisms. Now coming to this new chapter, Anatomy. What do you mean by anatomy? Anatomy is going to be nothing but the study of internal structures of the organisms is known as anatomy. In the last lesson, we have studied the morphology. Morphology means from outside, how a organism is looking from outside, it is known as morphology. Anatomy means within the body. When we are going to be cutting open the body, what are the structures that we are able to see within that body is known as anatomy. For example, suppose we cut open a human being, then we will be able to observe the different systems of the body, like the digestive system, the circulatory system, where the heart is located, how the blood vessels are running. All this we will be able to study when we are studying within the body of the organism. So that is going to be the anatomy. In this lesson, of course, we are not studying about the human being's anatomy, but here we are studying the anatomy of the flowering plants, that is your angiosperms. We will see in the angiosperms which are going to be the different type of cells, how the tissues are organized, how the tissue systems are organized in the case of the flowering plants. So to start with, let us first see the definition of anatomy. So as I have already told you, anatomy is the study of internal structure of plants and other organisms. You can see in these two diagrams over here. These are the transverse sections, that is the cut sections of the stem of the plants observed in the microscope. So here we can see the internal structures, how the cells are going to be arranged, that we are able to see in this diagram. Moving forward, let us go to the tissues. So what are tissues? Cells. Cells are the basic structural and functional unit of any living organism. When these cells are going to be found in group, or when these cells are going to be coming together to perform a particular function, they form the tissues. Tissues is nothing but group of cells is called tissues. So here we can see that tissue is nothing but a group of cells having common origin and function are going to be called as the tissue. So those cells which are having first of all common origin, they have to be from the same part and they should have a common function. Hence, based on the capability of the cell, that is what a cell can do, or based upon the capability of cell division, what type of division it is taking place, we can see that the plant tissues are grouped under two groups. The first one is the meristematic tissue and the second is the permanent tissue. So let us see in brief what is this meristematic tissue and permanent tissue, which will be the basis for you for the study of all the systems and the internal structures of the flowering plants. So first let us go to the meristematic tissue. The meristematic tissues are going to be those tissues which are undergoing continuous cell division. They are going to be actively dividing. Because of this meristematic tissues only, the growth of the organism is going to be taking place. So the work of the meristematic tissue is going to be only one thing. that is continuous division. They go on or they keep on dividing. So these are known as the meristematic tissue. These are the tissues where the active division and the growth occurs. Based on the position, meristems are being classified into three types: apical meristem, intercalary meristem, and the lateral meristem or the secondary meristem. So let us see these three types of the meristems, where they are present, based on which they are being differentiated. So coming to the first one, that is the apical meristem. So these apical meristems are going to be present at the tips of the roots and the shoots, and they are responsible to produce the primary tissues. So wherever there is going to be the growth of the shoot or the root, we can see that the apical meristems are going to be present. There are two types of apical meristems: root apical meristem and shoot apical meristem. So root apical meristem, which is going to be occurring at the tip of the root. So here the root tip that is going to be there, therein what type of the tissues are present, those are known as the root apical meristems. Whereas those which are going to be present at the shoot, that is which is going to be occupying a distinct most region of the stem axis, that is at the tip of the stem, are going to be called as the shoot apical meristem. Some cells left behind from shoot apical meristem are going to be forming the axillary buds. The axillary buds are going to be present in the axil of the leaves and can form a branch or a flower. So whichever are going to be growing on the top, they are responsible and they are continuing into the shoot. They grow, they increase the length of the plant and the new stem it goes on forming. But some of the tissues of the shoot apical meristem remain in some places, from where these are known as the axillary buds, from where a new leaf or a new branch or a flower can arise from this axillary bud. Then the second type of the meristems are going to be the intercalary meristems. Intercalary meristems are present between the mature tissues. So apical meristems are present here at the tip and at the axillary buds, and intercalary meristems are present in between the mature tissues. So in short, you can say at the internodes. Here we can see this is going to be the intercalary meristem. Matured, all these tissues have got matured, and here these are going to be formed or found in these mature tissues. Especially, we can see a specific character of the intercalary meristem in the grasses. So what happens in the grass? The herbivorous animals like the cows and goats are eating the grass, because of which the leaf of the grass is getting cut. But because of this intercalary meristem, that grass part is able to regenerate. So they occur in grasses and regenerate parts removed by the grazing herbivores. Then we can see that the apical and the intercalary meristems are primary meristems because they appear in a plant life and contribute the formation of primary plant body. During that, specific regions of the apical meristem produce dermal tissue, ground tissue and vascular tissue. So they are helping to differentiate the other type of tissues, we can say. Then coming to the third type of the meristem is going to be the secondary meristem or the lateral meristems. The secondary meristem or the lateral meristems are responsible to increase the girth of the plant. Girth of the plant means nothing but the diameter of the plant, we can say. So they are going to be present in the mature regions of the root and the shoots. They are usually cylindrical meristems and they are going to be seen only in the gymnosperms and the dicotyledons. The large trees you can see which are having a very large stem is going to be only in the case of gymnosperms and the dicotyledons. Monocots like your maize or maybe your sugarcane, they are not increasing their girth or diameter. They are remaining thin. The stem is remaining thin because therein the lateral meristem is absent. Only in the gymnosperms and the dicotyledons we can see that the lateral meristems are present. They are also known or they are also responsible for producing the secondary tissues. That is secondary growth they are known to form. Examples are going to be the fascicular vascular cambium, interfascicular cambium and cork cambium. Then coming to the second one is going to be the permanent tissues or mature tissues. We saw the meristematic tissues which are going to be having the one and only function, that is going to be continuous cell division. No other work they are performing. These permanent tissues are going to be the tissues which are having their particular work. They know what to do. They are been differentiated. So these are going to be called as permanent tissues or the mature tissues. The cells produced by primary and secondary meristem becomes structurally and functionally specialized and lose the ability to divide. So these meristems only are getting converted into the permanent tissues. They stop dividing and they take over the function. They start performing the function. Then they are called as the permanent tissues. So they are called permanent tissues and they constitute the permanent tissues. They are called permanent cells and they constitute the permanent tissues. So there are going to be two types of permanent tissues, simple permanent tissues and the complex ones. Complex we will be studying in my next video. Today let us see the simple ones. The simple permanent tissues are going to be those tissues which are having all cells similar in structure and function. The cells are going to be present, many cells coming together are tissues. So the permanent tissues are going to be having similar cells or the same type of cells which are forming a same structure and having a same function. So that are known as the simple tissues. So there are three types of the simple tissues: parenchyma, collenchyma and sclerenchyma. Let us see the first one, that is parenchyma. So parenchyma, it forms the major component within the organs. Most of the parts are made up of parenchyma. The cells of the parenchyma are having a isodiametric shape, which means they may be spherical, spherical is round, or they may be oval, or they may be round, or they can be polygonal or elongated. Polygonal is having 5 sides, or elongated means they can be like this, elongated structures. So they may be having a varied shapes according to where they are present. Their cell wall. Cell wall is going to be the outermost covering of the cell. The cell wall is going to, their walls in fact are going to be thin and they are made up of cellulose. Cellulose is a type of carbohydrate. And these cells are going to be closely packed or have small intercellular spaces. You can see over here, the cells are going to be present with small intercellular spaces in the middle. So these are known as the parenchyma. The main functions of the parenchyma are photosynthesis, storage and secretion. Coming to the second type of permanent tissue, that is the collenchyma. Collenchyma, it occurs in the layers below the epidermis in dicot plants. Epidermis is the skin layer. Outermost epidermis is the outermost skin layer. So below that, so as you can see here, this brown color thing is going to be the epidermis. Below that, this structure is going to be the collenchyma. It is found in a homogeneous layer or in patches. Homogeneous layer means a straight layer or patches here and there. It consists of cells with much thickened corners due to deposition of cellulose, hemicellulose and pectin. In this case, the intercellular spaces are absent. In the parenchyma, we could observe the intercellular spaces. But in the collenchyma, the intercellular spaces are not present. Instead, we can see that they are going to be having some kind of thickening which is going to be tightly bonding the cells with each other. They are having the thickened corners which are made up of cellulose, hemicellulose and pectin. Then, these collenchyma can also be of variant shapes like that of parenchyma. They can be oval, they can be spherical or polygonal, and they are often containing the chloroplast which is going to be having the photosynthesis pigment. Coming to the functions, these collenchyma are providing the mechanical support to the growing parts such as young stem and petiole of the leaf. As they are going to be having the thickenings, they are giving a mechanical support to the young stem and the stalk of the leaves, that is your petiole. The cells that contain chloroplast assimilate food, that is they gather the food. Coming to the third type of the cell, that is the permanent tissue, is going to be the sclerenchyma. The sclerenchyma consists of long, narrow cells with thick lignified cell walls having a few or numerous pits. When I am talking about the parenchyma and collenchyma, they are having a cell membrane within which the cytoplasm is present and nucleus is present, which is making them the living tissue. It is a living tissue, the parenchyma and collenchyma. But the sclerenchyma are going to be the dead tissues. So they are dead and they are without the protoplast. That is, there is not going to be any cytoplasm nor any nucleus is present. So they are going to be the dead tissues. Then what they are containing? They are containing only the walls, as you can see here and here. Only the narrow cells are present with thick and lignified walls having the pits. Based on the form, structure, origin and development, sclerenchyma are of two types, fibers and sclerites. What are the fibers? So fibers are going to be thick-walled, elongated and pointed cells generally occurring in groups. So these are going to be the fibers. To tell you in simple terms, fibers are nothing but if you have seen a coconut, while you are when you break open the coconut, that is remove the top layer, the brown color fibrous thing that you can see or thread like structures that are present, that are nothing but made up of the fibers. The second one is the sclerites. So sclerites are going to be, these are spherical, oval or cylindrical, highly thickened dead cells with a very narrow cavity is known as lumen. So this central part is known as the lumen. They are found in the fruit walls of nuts. If you are taking your nuts like your ground nut or your cashew nut or your badam, what we call, all these nuts are having a covering over them. They are made up of this sclerites. Also, in the pulp of fruits like guava or peru or pear or sapota, we can see they are going to be present patches of the sclerites. Even the seed coats of leguminous plants are nothing but your dals, what you have, tuar dal or moong dal, they are having a darkened color structure, seed coat they are having. So that is going to be nothing but the sclerites. And even in the leaves of the tea, the sclerites are going to be present. The main function of this sclerenchyma, it is going to be to provide mechanical support to the organs. So remember, parenchyma and collenchyma are going to be the living tissues. Parenchyma is known as the living storage tissue. Collenchyma is known as living mechanical support giving tissue, whereas the sclerenchyma is known as the dead mechanical tissue. So these are going to be the three types of permanent tissues that are present in the plants, that is parenchyma, collenchyma and sclerenchyma. In my next video, we will study about the complex permanent tissues.